started. So welcome to Fertility Planit. For those of you that this is your first session, we're so happy to have you and that this exists, the first ever conference about this topic, and we're just so excited to have this groundbreaking experience together and for everyone to connect, educate themselves, empower themselves, meet people, hear from experts. So welcome, welcome. So the topic today, as you know, because you're here, is genetic diagnosis and its impacts on your pregnancy. And we have a wonderful set of experts here. I'm going to introduce them. We have our moderator. This is Dr. David Hill. Dr. Hill is the co-founder and scientific director of ART Reproductive Center. Dr. Hill's career demonstrates a continuous series of achievements in improving pregnancy rates through applied research, as well as an exceptional ability to recognize and adopt new technologies that have promise for improving fertility. Thank you for being here. Awesome. Then we have Dr. Scott Sills. Dr. Sills is a physician at Pacific Reproductive Center in Irvine. Dr. Sills' research on advanced reproductive technologies has been presented before expert audiences in Africa, Asia, Europe, India, and North and South America, as well as the Middle East. Wow, we're happy to have you, Dr. Sills. And then we have Dr. Daniel Dumesic. Dr. Dumesic of UCLA Health System has clinical and research interests that include female infertility, in vitro fertilization, and polycystic ovary syndrome. Dr. Dumesic is a leader in several internationally recognized organizations and has received innumerable awards. Thank you for being here. And then we have Dr. Santiago Munne. Dr. Munne is the founder and director of Reprogenetics. Dr. Munne's research interests include developing new PGD techniques, and for those of you who don't know, PGD is pre-implantation genetic diagnosis. So it's looking at the embryos before they are implanted. Very exciting. Understanding the impact of chromosome abnormalities in human reproduction, as well as the production and study of genetically defined stem cells. So thank you for being here. So we have a wonderful panel today. I just want to remind everyone, we also, in addition to the speakers and this session and the other sessions, we do have a wonderful exhibit hall, which is across from the registration that you went through, out the glass doors a little bit outside there, but directly across from registration. Please go and visit those booths, because a lot of our speakers have booths there and you can talk to them more extensively, and there's a lot of exciting things going on in our exhibition hall, so we want to invite you there as well. And so now I just want to turn this over to Dr. Hill.
All right, thank you.
Oh, and one more thing. Hold your questions at the end. We will have a Q&A, and we're excited to have you interact with our speakers at that time.
Right.
Thank you.
Plenty of time for that. Well, welcome. Nice to see everybody here, and hopefully you'll get a chance to learn some things today. And believe me, I'm sure I will too, from our panelists. And without further ado, the way we do this is there's going to be about 5 to 6 minutes for each member of the panel. I'm going to ask them a question, and they're going to talk to you about it. And some of them have a few slides to support that. And then we'll move on, and then there'll be plenty of time to raise your hand and say, well, what did you mean by that? Okay, so, and any and all questions are welcome, of course. So we'll start with Dr. Sills, who you were introduced briefly to him. And it's funny, I've known Dr. Sills personally, and I won't dwell on it, but this is the first time I've actually met him in person, and an impressive fellow he is indeed. And he has a wonderful son with a great hat over there. But anyway, so a question to you, Dr. Sills. What is genetic diagnosis? Why is genetic diagnosis necessary or helpful? And for example, why should a doctor or a patient turn to genetic diagnosis, and what decisions and choices can doctors and patients make based on information and the results received?
Thank you very much, Dr. Hill, for moderating this august group, and I feel like I'm on National Press Club. That was a compound, complex sentence. I'll do my best to cover all the territory that's embraced.
Yeah, 10 words or less.
10 words or less. But thank you for coming. I hope you'll find this enjoyable. Can I get my first slide, which will help segue into my answer to Dr. Hill's question? The real focus of genetic diagnosis with clinical IVF is to help us help patients achieve their goal of having a healthy pregnancy. As my slide here is summarizing, the biggest factor in estimating reproductive outcome really is egg quality. And egg quality ramifies very directly on embryo quality. And the higher frequency of error chromosomally has been known for many years to be directly connected to maternal age. This is the reason why in the early days of IVF, multiple embryo transfers were necessary. Having to put 2 or 3 in to get 1 to take was sort of the mantra of IVF from the late 70s up until very recently. So what I'd like to talk about at this point is how getting a genetic fingerprint, if you will, of an embryo can help doctors select which embryo can be transferred so that we can only transfer a single good embryo. And that's going to really do a lot to attenuate the rate of twins and triplets, which is so common with IVF. Here's a picture of, and I apologize, I have about half of my practice is male factor, so this is actually a male karyotype, but the point of the slide, it can still be made in that every cell in the body has a slide like this that can capture genetic information. And this is the beginnings of our understanding about the chromosomal mix-ups that can happen in human biology. These little arrows, red arrows, show where there are mistakes, serious errors that have been introduced through either a hereditary process or perhaps an environmental process that had a very serious downstream effect on that individual's ability to make sperm. When we look at ways to tell about embryo cells at the time they're in culture, we wanted to have a technique to identify chromosomal competencies in cells obtained from human embryos before they're transferred. The way that array comparative genomic hybridization does that is probably outside the scope of this afternoon's gathering, but suffice it to say that it's a very highly precise, very high resolution way to put a caliper on the chromosomes to measure any genetic gain or loss of material. And what we can find out from our work at Pacific Reproductive is even though the embryos may look pretty on the microscope stage, they can still harbor very severe and very profound chromosomal mistakes. Later on, the other guests will probably talk about aspects of embryo testing, so I don't want to overlap any of their content, but I'll just say in brief that our clinic's experience with array CGH enabling fresh embryo transfers is really made possible by doing a biopsy at day 5 at the blastocyst stage with trophectoderm cells, the cells at the periphery of the embryo being sampled, and then 12 to 13 hours later being able to make that patient able to receive a fresh embryo transfer seamlessly. That helps patients not have to wait for a frozen cycle later. This photograph here, I hope, can illustrate the...
Can I interrupt you for 1 second? I just want to adjust your microphone because we really want to hear you, and it's going inside your jacket.
I'm glad you're here.
Yeah, because you're saying such important stuff, you know, we want to hear you. Thank you.
Testing 1, 2, 3. Okay. So in this picture, to the... I'm sorry?
Much better.
Oh, thank you. We'll have discs available at the end of the concert at the back. But the embryos that you see here look very, very good. They're hatching blastocysts. These photographs were taken at about day 5 in the IVF lab at our Irvine office. Now, the problem is that even though these look very, very good, when you take a look at how they perform at the chromosomal level, they are very different. If we don't do a DNA fingerprinting of embryos, that embryo on the bottom has an entirely missing chromosome 12. If that embryo were to be transferred, either it would not implant at all or possibly temporarily implant and then miscarry at 9 or 10 weeks or something like that. Not a good outcome. So just doing a beauty contest of embryos based on their physical appearance is clearly not the way forward. And what I think we're all aiming for is how to make the best of the great scientific advancements that are being racked up every 6 months or so in scientific journals. The way it's helped our patients has been noticeable. If we look at the embryos the old-fashioned way and just transfer one embryo at a time, pregnancy rates will come in at about 45%. Using a DNA array that can be significantly lifted to about 70%. And importantly, there are no twins. If you do a single embryo transfer, the only way to have a twin outcome is if the embryo splits, which it's controversial as to whether IVF increases that rate. I don't think that it does, but that would be identical twinning, which is very rare. So I think this could be a way forward in terms of helping get pregnancy rates high, keeping multiple gestation rates low, and this also has the advantage of doing screening assessments for things like Down syndrome, Turner syndrome, Klinefelter syndrome, all the other chromosomal errors that are of interest to our patients. So with that, I will pass the baton to Dr. Dumesic, and look forward to your questions later. Thank you.
Thanks very much. So the question for, and I can tell you at this juncture, as the lab guy who has had to resort to the beauty contest of looking in the microscope and telling and kind of going, well, this one looks good. Let's use that one. No, that one looks good. So let's throw another one in there just to be safe. This stops that. So it really, in my mind, is the way forward. I can tell you from my perspective. So the question I have for my good friend and colleague, Dr. Dumesic, is this. How does genetic function impact a person's fertility and chances of conceiving? Can you give an example or elaborate on what a patient can do to improve genetic function of their gametes? And how important is it for patients to understand genetic diagnosis and function and what goes on in the laboratory?
Okay. Well, thank you, David. I also like to thank the speaker or the organizers of this meeting to have me come here and be with these fine colleagues. And I'll perhaps elaborate in a slightly different way in terms of function rather than the actual identification of the genes themselves. And I'd like to maybe start by sharing with you a little bit of a historical perspective going back to 1651, believe it or not, when William Harvey, who happened to be the physician for King Charles I, spent a lot of time in the fields during the hunting season when they were hunting for the fallow deer, which you actually see right here in this particular image right here. And he was very interested in looking at the time, the ovaries, which they didn't understand their function at all. And over 100 years before the egg was identified, he came up with the idea that the egg, there must be something called an egg or equivalent thereof that fertilized and led to the beginning of life. And this was pretty remarkable because the microscope hadn't been invented, the egg had not been discovered, and people didn't even know what the ovary was. So one of the last publications he made was on the origin of the generation of animals, and he had in his book this frontispiece of Zeus sitting on a pedestal here holding an egg with the inscription in ovo avia, basically saying everything comes from the egg. And here you see the human being, you see the fallow deer that he made this theory on, and all of the other species. So as we're talking here about the high technology of what's happening in 2013, I'd just like to again emphasize we're sitting on many, many shoulders of giants that really led to all of this technology. I thought it was amazing. These people could figure this out when they had no idea of what they were looking at and just used inference. So with that being said, I'd like to move to my topic, which is really looking at the function of genes. And as Dr. Sills has pointed out, a lot of the aspects of the embryo originate from the sperm and the egg. And particularly when we do these retrievals after stimulations and retrieve the egg, it's enclosed by several cells which provided energy, which are called the cumulus cells. And the important aspect that we like to look at is really the function of these genes, because potentially we can do something about the management of these functions if we understand them. Here's a good example of several metaphase 2 eggs, mature eggs, that we would have normally used in the lab in clinical practice, but as part of a study that we were doing a few years ago, people volunteered up these eggs for research. So we are able to map all of the genes of the human egg. Now, with that being said, these genes are normal in terms of identifying them, but the question we looked at was really their function, their expression. Are they overactive? Are they underactive? And what we found in these particular women who happen to have polycystic ovary syndrome was that if you looked at the normal eggs here, they would fall into one group, and this is a map looking at all of the genes and their pattern of expression. The normal women have a certain pattern, and the women with polycystic ovary syndrome have another type of pattern, despite the fact, as alluded to by Scott, that these eggs look totally normal under the microscope, and yet something is wrong with the function of these genes, or I should say different, perhaps, maybe rather than abnormal. And what this slide doesn't necessarily show well, because I think of the lights, but one of the things we're interested in is fat metabolism. And the reason for that was when we looked at these genes that were abnormal in expression, many of them were driven by substances which control energy utilization, particularly fat and lipid and insulin, which you've probably all heard about some way or another through the media as implicated with type 2 diabetes and a number of aspects of being overweight or metabolic dysfunction. So we were very, very interested in that in particular. What this slide doesn't show too well because of the lighting is that as women's body mass index goes up, which is a measure of how heavy they are, what happens if you look at the follicular fluid, the amount of insulin in their follicle goes up in proportion to their body weight. And this makes sense because the follicular fluid is comprised largely of fluid coming from the blood into the follicle. And so if those hormones are able to change and alter gene function, even though the genes are normal in configuration, then you're going to have some abnormalities to function as well. One example of that where we've tried to pursue a little bit further is to actually map out within these cumulus cells the amount of lipid or fat within these cells. And the reason we started doing that in collaboration with Dr. Hill is that we recognize that lipid or fat in cells has a function. It's important for hormone production. It's also important for energy utilization because these fat metabolites are used for energy so that the egg can do all of the things leading to an embryo in its development that requires energy utilization, which gets into aging and some other aspects. And this is a good example of, if you look at the cumulus cells, you see that the greenish-yellow configuration here that you're seeing in all of these cells is actually lipid that we've been able to measure and quantify. The blue dots you see here are the nuclei, which demonstrate that these cells are actually intact cells. They're not remnants. They're not some abnormal lipid that's just floating around. So we're very interested in looking at gene function and the types of things in the environment that we can control, whether it be body weight, whether it be some of the hormone levels that are abnormal in some of the follicles of individuals, all of which we hope we can control as clinicians so that by the time we get the eggs that we can hand over to the laboratory, not only the gene function, the gene expression, but the gene function are both as optimal as possible for the laboratory people to work with. So I think that's all I had to comment about.
At this point.
At this point.
Thanks very much. Our third speaker is my wonderful friend, mentor, and colleague, Santiago Munne. who, again, I remind everybody here that his company, Reprogenetics, is sponsoring this session. Santi, a question that I would have for you. Could you tell the audience about the latest and cutting-edge advancements that we have in genetic diagnosis, for example? What kind of information is becoming available to doctors, to patients from the laboratory? How can this information and diagnostic evidence impact decisions around fertility treatments, conceiving, and pregnancy?
Thank you, David. Well, this information comes at two different levels. One is to testing couples to see if they have genetic diseases or mutations that could impact their reproduction, or also impacting producing babies that have genetic diseases. And the other, it's at the embryo level. So right now, through the Genome Project, it's been possible to sequence all the DNA of the human body. And we know that there are 3,500 million letters in the genome. Those are organizing genes, and there are about 35,000 genes. And these genes, they could produce genetic defects in babies. For instance, just one letter in one of these genes could produce a genetic defect, for instance, cystic fibrosis, mutation delta 508. It means that the letter 508 in that gene is missing, and this is producing cystic fibrosis. So right now, what it's possible to do is to screen from hundreds of gene defects at the same time with very affordable tests. Usually they are covered by insurance. For instance, one of them can detect for 178 diseases, and I've done it myself. I have 3 of them. The interesting thing is that everybody in this audience probably is carrying some genetic diseases. It's calculated that about 14 to 18 mutations are found in everybody. The question, or the important part, is not if you carry a genetic disease, is that if your partner carries the same one. So as I said, everybody is a carrier of at least 14 to 18 of them. And what's important is that your partner is not a carrier of the same one. So now there are tests in the last 1-2 years that can test for many of these genetic diseases. And I definitely recommend that you do it because of the risk that it could pose if you don't do the test. On the other level, and what's going to happen in the next few years is that we're going to be able to detect not only these 100 and 200 diseases that we are testing right now, but 6,000 or more that are known at this point. On the other level, and Dr. Sills already mentioned this, at the embryo level, if we find out that you are carriers of genetic disease, or both partners are carrying of the same genetic disease, then we can go to the embryos and take a small piece of the embryo. It doesn't damage the embryo at the blastocyst stage, and then do a genetic analysis and select the ones that are normal, so you won't have an affected baby. This technique, which is called pre-implantation genetic diagnosis, can also be used to test the embryos for chromosome abnormalities. These are not inherited, so you are not carriers of it, but chromosome abnormalities increase dramatically with advanced maternal age. So a woman that is 35 will have at least 50% of the embryos chromosomally abnormal. A woman that is 40 is going to be close to 80%. So we could do the same thing, go to the embryos, do a small biopsy, test it genetically, and if they are chromosomally normal, then we'll implant those. By doing that, you do 2 things. One, you reduce the chances of the embryo miscarrying if they take, and also the ones that are abnormal, usually they don't implant or miscarry. Therefore, you have a higher chance of getting pregnant and getting pregnant soon. The only downside could be that we won't find any normal embryos. And this could happen, again, with advanced maternal age. But if we find one that is normal, what we found recently is that an embryo that is normal at any age, they implant at the same rate regardless of the age of the mother. So what's important is not how old are you, but to find that normal embryo, and then they all implant at the same rate. The only problem with advanced maternal age is that you have less and less embryos, and if you don't test them, then you are choosing a random one of them, and you are replacing more and more abnormal ones, and that's why the pregnancy rate decreases with advanced maternal age. But if you find a normal one, then they implant all at the same rate.
Terrific. Thank you. So everybody has abnormalities. I guess that's where that expression, nobody's perfect, comes from. Anyway, well, very good.
If you don't mind,
go ahead.
I wanted to point another thing, and that this is the same for egg donors. So the paradigm right now is that when you go to an egg donor or a sperm donor, they will tell you that, well, we've done the screening and these donors are perfect. And there is not a single donor that is perfect. And the only way around it is to test the donors for all these diseases and test the recipient and then try to see that they don't have the same thing. Then that's fine. But don't buy that there are perfect donors.
Very good. So we have a few minutes here. I have some questions that I can ask of the panel, but we have a couple minutes for anything out of the audience. Remember, there isn't anything, there's no silly question. And we can provide you with a microphone, or you can just shriek it out. Yes, ma'am.
It needs to be on mic so that we get it on the live stream.
Ah, okay.
I just wanted to get some more information about, you had mentioned that it's unlikely that the egg will implant if there is a chromosomal abnormality. So what are the percentages of that egg actually implanting if there is an issue? And once it is implanted, other than amniocentesis and CVS, where are we at technologically to be able to screen and find out what is going on and what options are open at that point?
Yeah. So if it's abnormal, usually they will not implant or they will miscarry.
Usually 70%.
Okay. The rate of miscarriage goes up to 40% when you are 40, and it goes from 17% to 40%. All the others miscarry. You could do two things. Obviously, the best thing is to do a pre-implantation genetic diagnosis, to do the biopsies in the embryos, and then the rate of miscarriage goes down to, I think, between 3 and 5% with the new technique. If you didn't do the test, what you could do then is prenatal diagnosis, and now there are some tests that are non-invasive, so it's just a blood test, and they will detect if you have the chromosomal abnormalities that can reach term, so trisomy 21, which is Down syndrome, and 3 or 4 more. But then it's too late. You will have to decide if you want to carry this abnormal pregnancy or to terminate. So obviously, if you are conceiving through in vitro fertilization, probably it's much better to be able to test the embryos and then just have a normal embryo transfer.
Over here.
Where's the research on if treatment before IVF or during the IVF cycle with growth hormone enhances the DNA repair mechanism? Is that true, or is that just
Either Dr. Sills or Dr. Dumesic do that one.
Well, that's a very good question. Growth hormone is a substance that comes in and out of vogue in the neighborhood of IVF every so often. Dr. Dumesic may know more about this than I do, but my first encounter with publications specifically focused on growth hormone were in the late 1990s. And there were a few studies that looked at the pregnancy rates when growth hormone was used adjunctively with traditional gonadotropin stimulation regimes. And there really was not a significant difference in terms of pregnancy rates or miscarriage rates. The problem came into the equation at the economics level. Growth hormone is not easy to get. It's a substance that used to be available only cadaverically. Now it can be genetically engineered, but previously it was obtained from pituitary glands of people that had passed away. The primary target clinical audience for growth hormone was in a pediatric population. And if a bunch of patients who were doing an elective fertility treatment were consuming all the growth hormone, then there wouldn't be enough for the babies and kids who needed growth hormone for other clinical indications. This became a bioethical issue in our field. It was put to rest, I think, by the fact that the data really were not very encouraging in terms of did it really boost outcomes.
Even with advanced maternal age?
Well, advanced maternal age, the incidence of growth hormone deficiency in those patients is low. So if you're going to supplement something, I would always like to tell patients I want to be able to prove to them that I'm giving them something that they truly need. I think there probably are some programs that operate on an experimental basis. It is, I think, still considered investigational. But I don't look at growth hormone as being something that is likely to enter the mainstream of clinical IVF unless there's a specific clinical circumstance that would warrant it.
Can I comment
Yes,
to
sir.
that? I think a lot of the things that happen is we initially treat everybody with something, and then it turns out there's subpopulations where it might be helpful and others, so across the board it isn't, and you sort of lose your effect until you figure out exactly who needs it. I think the paper you're probably referring to is Jan Tesaric's work in advanced maternal age with growth hormone has been applied. And he showed in his study that there was an enhancement for pregnancy outcomes. So it's controversial. The things that are interesting about it are that when we think, as endocrinologists, we think about hormones that, that go from one place, presumably in the blood, to the follicle to do something. And in order for that hormone to do something, you have to have a receptor for it on whatever cell you want it to work with. Since the original work that was done several years ago with growth hormone across the board, it has been shown that growth hormone is present not only in the cumulus cells, but in the egg itself. So now you have a logical connection where there's plausibility. Also, growth hormone affects lipid metabolism, and it affects glucose metabolism. So you have the underpinnings to put the biology together to make it relevant if you know who it's relevant for. So I think from my perspective, I agree with what Dr. Sills' statements are. I personally use it, but we try and limit it to people that we think it's important to, provided we understand the biology of it. And that's what we're trying to figure out. So I think these hormones, and a lot of them are like that. Dexamethasone is another one that's kind of come and it's gone and it comes back again in different ways. Because as you learn more information, things come back on the table again. And then you say, well, okay, maybe this group of people who really can benefit is quite narrow, as opposed to just give it to everybody. And I think that's how most of the field moves forward. So I think there's strong merit for it. There's probably some people that benefit from it. And there's a proof of concept in the sense that there are receptors on these key cells that see it, which means there might be a way that it's beneficial.
Thank you.
Regarding the genetic testing, is that a standard in the field? Does every endocrinologist or lab do it? Is it at the same level? Does it depend upon a specific recommendation, or does the patient opt not to do it? How does that work?
Dr. Munne, you want to take that one?
Sure. We received samples from about 150 clinics in the US, and there are 300, and probably there are a few other labs. So I would say that PGD is now a standard of care in patients that are over 35. At least in the majority of clinics, they will consider now standard of care for the amount of cases that we see that are being done. Regarding testing the couples for gene defects, The standard is to test for a few of them, but I think these guidelines are very old because it was before the new technologies appeared. And if you are going to test for 10 diseases, why not test for 200 diseases if it's the same test?
So what your organization would do may be different than what another organization would do?
There are some guidelines from the American College of Human Genetics and others, but they have not been renewed lately. So.
Thank you.
So hi, thank you so much. I'm really enjoying this. It seems like a no-brainer if you could test for all these diseases and you'd want to, find out. And I guess it's a twofold question is, what's the downside of doing the testing? Like, is it controversial at all, or should everybody be doing PGD? And then the other side is, if, for example, like you said, there's no perfect egg donor, and I'm looking for a donor, and I test her, and she comes up with these weird mutations I've never even heard of, and now I'm petrified because I don't, and my husband doesn't want to be tested, or I don't want to go to the expense of testing my husband. Well, where am I at? Then I'm just paranoid now because I've just done this test of 173 mutations, and she's got 2, and one of them is maybe 1 in 25,000, but do I really want to take that chance? So to me, just logically, is it a double-edged sword, or really, and I guess also, how much is it? How much more expensive?
Yeah, so it's $300, so it's not too much. $300, $350, something like that. Yeah,
what is that specifically for the 300? Is that for the PG?
The thing is that we have to change the paradigm that we had before. Before you go to an agency and they would tell you, yeah, we'll find a perfect donor for you. And they will test cystic fibrosis, fragile eggs, and a couple more. And what happened is that there are 6,000 of them. And if, as I said, I test myself, I test my wife, we have different mutations, and luckily our babies are normal, and according to our eyes, above average. But it would be really. I think we have to change the paradigm because this tool is available, and the only thing that you have to do is to make sure that the donor and the other partner are compatible, and then you reject less donors. Right now, we're rejecting probably 10-20% of the donors because of these abnormalities, because we're testing only one. If you test all of them, you just change donors, and then there will be a good match for somebody. So as an agency or an egg bank or a sperm bank, you wouldn't reject anybody because of that, provided that the recipient or the other partner are compatible with them. You don't have to reject them. You just have to choose one that are compatible. That's the new paradigm, I think. It hasn't reached yet. It's still taking some time. Because there are interest in that they are selling this concept that will find a perfect donor for you. And well, now we know that each one of them have 14 to 18 mutations.
It also probably has to do with kind of in any field, as you know, there's kind of the status quo. Well, we've been doing it this way for a long time. And to get the bulk of the opinion in that field to move in a new direction, that's where we are right now. As Dr. Munne pointed out, at least a third now, if not half, of all IVF clinics have embraced this concept of improving patients' chances for having a healthy term delivery by using this technology. Does it cost more money? Yes, it does. It adds significantly to the cost of if you're going to do in vitro fertilization and embryo transfer. There's no question. But the trade-off pales in comparison with having to, if you don't do it, let's say you don't get pregnant, you got to do another IVF try all over again. And the amount of information that you can gain just from doing these evaluations is, as I see it, from my perspective, was a non-clinician perspective, but from a scientific standpoint, it makes a lot of sense. So are there... question back there.
I think you mentioned, Dr. Munne, that if some clinics, the standard of care is that if you're 35, that they do the testing. So if you're under 35, should you insist, if you're going to do IVF, that they do genetic testing?
I think Dr. Sills can answer this because they have an amazing paper on young patients, and they show that it works equally well. Or
That's a good point, and I think that it does show the paradigm shift that was alluded to earlier, in that as we find comfort and familiarity with a particular diagnostic test, especially something that's as fundamental as chromosomal integrity, the initial populations that it was applied to, recurrent miscarriage patients, older patients 40 years of age and above, those who had a history of recurrent pregnancy loss for whatever reason. The use of PGD was profoundly helpful for those patients. In May of last year, our group published a paper on applications of embryo biopsy and testing among under 35-year-olds, first-time IVF patient, and no history of miscarriage. These should be very robust patients that are very low risk. And when the spreadsheet of their chromosomal integrity was put before our computer, the error rate was over 40%, which was shocking. And it began to shed some light on why it's so important for patients who have embryos selected randomly on the basis of appearance. 2 embryo transfers, and sometimes even 3 embryo transfers, end up as a singleton delivery, because the misfire rate, so to speak, in embryos, even if they look outstanding under the microscope, can still have some very tragic problems. Now, the question about has this made its way into the top shelf status of tests that should be embraced for standard IVF, I talk about it with patients irrespective of their age, but I think at the end of the day, it is a personal choice. It is costly. It's sometimes covered by insurance. I like to write them letters and challenge them when they deny this, because I think it's a no-brainer. It's an investment in disease. It's the ultimate in preventive medicine in terms of trying to remove something that can cause severe handicap or disability later. But it's something that I hope that as more forums like this happen and the exchange of ideas happens, that there'll be a public expectation that this sort of technology should be talked about more, so that it will help improve patient outcomes.
As someone who's done a couple of rounds of IVF and had miscarriages and was fortunate enough to have one of my miscarriages tested for the chromosomal damages, one of the things I really didn't come away with, and I don't know if that can be done with PGD, is can you take your findings or the learnings from the testing to help you with the next round in terms of what can be, should you even try this again, or what would you do differently the next round? And that would really be helpful to many of us, I think, who are going through this process.
Yeah. Did you find out if what was the result? Or?
Yes, but I can't remember. Like chromosome number 46. What? Chromosome number 16 was too many.
Trisomy?
Trisomy.
Okay. Close enough.
That's sporadic.
She had 3 copies, and we're supposed to have just 2 copies of every one of our chromosomes, and if you have 3, it's an extra dose, and not good. So leads to miscarriage. And so your question is, okay, so what happened this time? What can I do with that information? Is that about right? Okay, Dr.
Yeah, so the data shows now that if you had the previous spontaneous abortions and it was chromosomally abnormal, like in your case, the trisomy 16, then you have a much higher chance of producing the same event. Not the same chromosomal abnormality, but another one. And when we do PGD, the rate of miscarriage goes from 48%, I think it is, to about 5%. So it's very, very effective when in this instance.
You just wouldn't have implanted them to begin with.
Yeah. Yeah, exactly.
So I just want, for clarity purposes, if I'm deciding now I'm going to go to an IVF clinic, and this is not something that they're already standardly doing, is it something that when I'm at that clinic, I ask them and I opt in and they send it? Or do I go to you first and then somehow take my eggs that look good from you back to that clinic? What is this process? Because it seems like there's a gap in the conversation.
You go to the clinic.
And then I ask them specifically to send it to you, and I pay them directly for this to go to you. Yeah. And this is a standard option at all of these clinics that's just maybe not known about? Or do I have to find a certain clinic that does do that already, that's on your list or something?
As I said, we work with almost half of the clinics in the US, and there are other labs that work with the rest. So all clinics probably have one lab, and some have the lab inside. So I think it's readily available. It's just that some doctors, for instance, for younger patients, it's not the standard of care yet to offer it. That doesn't mean that it doesn't work. I think it does work, and it, as Dr. Sills demonstrated, it does work. And it could be recommended to everybody, but it still hasn't reached the level that everybody's doing it. So maybe you have to ask. But for others, let's say recurrent miscarriage, advanced maternal age, most doctors would talk about it with you and recommend it or give you the option.
Maybe this will help a little bit. So when it's determined that you would benefit from in vitro fertilization, and that maybe in the course of the evaluation of the couple, let's say it's discovered that maybe you had a miscarriage in the past, and it was due, the products of that conception were analyzed, and it was discovered that it was due to a chromosomal abnormality. And frankly, that happens a lot in miscarriages. So if they find that, the clinic may say, you know, there is a pretty good correlation with, if this happens, that it could happen again. So let's try to prevent that. Let's also do pre-implantation genetic screening or diagnosis to make sure it doesn't happen. So you go through your in vitro try, which means you get a bunch of these injections, and then the eggs come out. The eggs are converted into these pre-implantation embryos in the laboratory. By about the 5th day of development, there are about 100 cells. We take just a few of those cells off, and actually, even by that time, we know that the cells we're taking are not the cells that are going to ultimately become the baby. They're going to become the placenta, other aspects of the fetal placental unit. So we snip off a little piece, and we send that biopsied material to testing laboratories such as Dr. Munne's. Takes them overnight to give us the answer, which comes back to us on a secure portal on the internet. And they'll say embryo number 1 is a female and normal. Embryo number 2 is a boy, not normal. Embryo number 4, and so and so and so, depending on however many embryos that you have. And we can either put them back, the ones that we now know are normal. We can do a fresh embryo transfer that following day, or what is gaining a lot of ground now is with the advent of what we call flash freezing or vitrification. It's a very, very effective way of freezing embryos. Embryo really doesn't even know it's been frozen. And so we then tell the patient after the biopsy, you go home, we'll get your results, and we'll find the results, and then you come back on a cycle that is not the one that we took the eggs out, because we're finding that there's a lot of stuff going on physiologically with you. Your estradiol levels are up here, and some of that stuff actually is not so great for the endometrial lining. And so we have you come back when that on a non-stimulated cycle where everything is calmed down, and all of the things being equal, the pregnancy rates are actually higher with these thawed, determined embryos to be normal by this stuff that's called CGH. So that's kind of where we're at. Just so maybe everybody in here knew all of that, but if you didn't, that's the way it's done. I see on our time thing that we've got, I think there's some online, Facebook, Twitter.
No, not at this time.
Not at this time.
I have a question, though, I'd like to ask. And thank you for that extra explanation. I appreciate that myself. I'm curious, like, the factor that I'm going to use the word quality of the eggs goes down with maternal age, right? What is that about? Like, let's say the person takes really great care of themselves, right? Woman A is 45, and look at her. She looks like she's 20. She's running around. She's got a lot of energy. She eats great food, organic, everything. Woman B, 45, eats at McDonald's every day. Like, you could say she's aging 45B, and 45A isn't aging the same way. So wouldn't it stand to reason that 45A's eggs would be in better shape than 45B's eggs? Or is it not about that? Because it's just like this whole thing, maternal age, eggs go down, the end.
Maybe I can give the first shot at it, and because that's a complicated issue, and then the other speakers can probably chime in. But that's a real. What you mentioned is very important, because people come into us and say, look, I'm in great shape, and therefore I must have good eggs, and that's really not true at all. The truth of the matter is some of these chromosomal abnormalities in terms of trisomy 3, monosomy 1, a misconnection, if you will, of the absolute number of a certain gene pair, is a complicated issue. But essentially, when an egg is mature, then those two pairs of chromosomes have to split and go to opposite sides as the egg gets rid of some of the chromosomes to allow the sperm in. And that's driven by these little microfilaments that pull the chromosomes to the sides, and maybe the guys will be a little more specific in a moment. But DNA is very sticky. So when those filaments break down as the egg ages, then those two pair can be pulled to the wrong side, or they can be pulled in abnormal directions. And that's an aging issue. Now, on top of that, the genes that control those chromosomes to split and separate is also driven by machinery that pulls these microfilaments to the sides, and that's driven by FSH. That's a hormone. So here again, you see this link between hormones and chromosomes coming together. We're struggling with that all of the time, because as many people know in the crowd, when people get older and they're undergoing stimulations, we're giving them more and more amounts of FSH to drive those follicles. And yet we know that's interacting with some of the machinery that controls the separation of these chromosomes. So the truth of the matter is, at this point in time, there's not a lot we can do about it, other than take those embryos and figure out which ones are chromosomally balanced or not. In the future, whether we can control that all remains to be determined.
So what you're saying is nothing has been researched about how to improve the filament function, if I'm hearing the words correctly, that is separating the DNA opening, right?
Well, no, I'm not saying that. I'm saying a tremendous amount of work is being done in research in that area. But effectively, at this point in time, as of 2013, what Dr. Sills and Dr. Hill and Dr. Munne are talking about in terms of saying, okay, we can't necessarily control that yet, but we can take those embryos and figure out which ones did it right and which ones did it wrong, and screen and select. Maybe 5 years, maybe 10 years from now, we'll figure that out. And I can give you an example. There's somebody named Bob Casper in Canada that's taking this on a metabolic level and saying, well, maybe the energy utilization to separate these things needs to be improved, because that's driven by mitochondria in those cells. And maybe we can kind of pump up the energy utilization and try and improve that, but that's all theoretical, although he's doing a study on that. So there are some opportunities, but right now what the speakers are saying is really what we have at our disposal.
And the growth hormone that was mentioned earlier for the older mothers, is that what it's going towards, that particular function?
Not particularly. The growth hormone is really looking at an issue we aren't even talking about, and that is the cytoplasm of the egg, because the egg has the genes in the middle, and the cytoplasm is what holds the energy organelles around it. The growth hormone and other hormones are looking at energy utilization of the cytoplasm, and that's really not what we're talking about. We're talking about the genes in the middle of the egg.
We have a question over.
So, if I understand correctly, so basically you have a new process for filtering embryos, right? A way of finding the best of the best. Isn't that going to make it more rare that you find the best of the best? Doesn't that actually reduce the number of potential embryos that are out there in the world? because you're basically eliminating all the ones that were maybes that people are using right now, and creating a field of just, we know these are all really good, but it's potentially a much smaller field. So they'll become more rare, become more expensive, and harder for people to get pregnant in the long run?
Let's look at it another way. Mother Nature, call it Mother Nature, call it wisdom of the body, whatever you want to call it, but if you have a couple that doesn't have any fertility issues, and they fall in love, they get married, and they want to have a baby, they actually only have about a 25% chance of it working every month. And that's not because she's not releasing an egg, and it's not because a sperm isn't finding the egg and fertilizing it, but things happen. And they just don't happen with perfect fidelity, and they, even under the ideal conditions of, and they're in their 20s, and he has an Olympian sperm count, and she ovulates like a clock, but the fact of the matter is that human beings are just built this way, that only about 25%. So this is why, after a year of trying, okay, if you're not pregnant, you really need to go and see somebody like these two gentlemen to my left, because fertility specialists can very quickly, with a few tests, home in on what's going on. Now, to your question about, well, if you're screening the embryos, you start out with a whole bunch from a patient, let's say 10, and depending on her age, and really, the chromosomal issues are oftentimes, pretty much the majority of the time, they're coming from the egg and not the sperm, and there's reasons for that. I know it's not fair, but it's the way it is. All we're doing is we're just doing what nature's done, and we're just going, okay, well, this one's good because we've screened it, and then we put that one back, and the odds of pregnancy, of course, go up, and the chances of miscarrying go down because we know what we're putting back.
David, can I make a point?
Please.
For some of us who are maybe older in the field, when
that would be
some of us more than others, when we first started many years ago, we didn't have these wonderful capabilities to grow the embryos out and check them, and that was just unknown. So what happened was, as soon as we made embryos, we froze them right away, immediately, almost at the pronuclear stage. Well, what that did is that caused a stockpile of extra embryos that we know we would have never used now in today's time. And what happened, and it wasn't too uncommon for people to have 15, 20 embryos frozen, and then every month you would go in and you would play all of these hormones and then transfer a couple, and that wouldn't work, and then another month you'd do some more. And it took forever. And the success rates were low, the expense was high, and the emotional trauma, which we haven't even talked about, was astronomical.
Huge.
So as we've moved forward in this field, and the gene people have gotten in here and really taken a very close look at this, it's really the efficiency of the process, because when people come in, the average age of our patient in our practice is about 36, and that's very common for most practices. People don't have time anymore, and they want to move on. So some of these things are a cost-benefit ratio. That's what's really the stumbling block. Is it really of value to do this? But I don't think anybody's being critical that these aren't wonderful things to do, so that you make the practice as efficient as possible for the couple. That's what it's all about.
We have a burning question here.
Go ahead.
I just have one question. What are the chances that doing a PGD will damage the embryo cells? Yeah.
So there has been now, I think, a quite definite study showing that if you do the biopsy on day 5 at the blastocyst stage, which there are about 150 cells or more, you are doing the biopsy on a layer of the embryo that will become the placenta. So that's a much better way of doing the biopsy than when we were doing it on day 3. On day 3 now there are some studies that there could have been some damage when you were doing at the 8-cell stage. There were only 8 cells, and they haven't been differentiated yet into different layers. Now that we are doing it a much later stage, not much later, 2 days more, but there are already 2 layers of tissue, and one is going to become the placenta. So it's like a very early prenatal diagnosis. You're taking cells from it will become the placenta, not the fetus. So really, you're not touching the fetus. And the latest studies show that there is actually no effect, and at the contrary, we are improving results. So
we biopsy hundreds of these embryos every month in our program, and it just doesn't happen. It just doesn't. It's extremely rare. If an embryo is biopsied and the embryo doesn't make it after that, there was something really wrong with the embryo. It's really, once the technique has been worked out, and it's worked out now that it really is almost a non-existent thing, that the embryo is damaged by the biopsy procedure itself.
Have you found any risks for the embryo itself in the long run, long-term side effects once the healthy child...
Take a little piece of it off and it comes out missing a foot or something like that? No, the answer is no. There have been studies, it's been around now a pretty long time, and there doesn't really seem to be anything like that. Certainly, I'm being facetious about missing a part of the body, but, could it affect intelligence? Could it affect, something? And we've yet to see it. There's a lot of IVF that's done in the world now. In fact, I think it's estimated in the United States that something like 1% to 2% of the population now is being added to by assisted reproductive technology. And the number is even higher in Europe, where they do, there's almost twice as much, maybe more, 3 times as much IVF that's done. We're always looking for subpopulations within patients that have IVF to see, are there groups that you see a higher incidence of birth defects and things like that. The risk involved in IVF and any of the ancillary things that we may have to do for certain patients is certainly not zero. But there's always that risk benefit. But overall, it's a pretty safe practice, what we see. Okay, so is it Twitter time or one more? Somebody, I see a few hands. Yes, ma'am.
I just had a question on gender. You had said earlier you can determine which embryo is male or female. Do you have a lot of clients that are requesting gender selection? Because I know that's a new hot designer thing. So
that does come up occasionally. It's not something that our program advertises, but it is something that is deeply important to my patients. And I think my consults take about an hour, so I talk a lot. So I typically cover the fact that whenever you get the report card, so to speak, of the embryo, as Dr. Hill said, one of the pieces of information that you glean from array CGH is embryo sex. That's going to be part of the quotient of the data that is delivered from our laboratory colleagues. So if they have an agenda about what they want to have transferred, I say if you would like us to control which of this panel of embryos that have checked out as being chromosomally normal, please let me know. And I think that's the opportunity for them to say, well, we'd like, if it's all the same to you, Doc, how about we just transfer embryos number 3 and 4, or maybe just embryo number 1. So I think there's been a lot written about elective embryo sex selection, the ethical luggage that accompanies that debate is extensive, and it's been unpacked by lots of authors in Europe and in America. I think that's still very contentious, but we could have a whole panel just on that topic probably to fully develop it. But you're right, the point you're making is exactly valid. It is something that is knowable. That's something that was knowable from the earlier FISH applications of the earliest days of PGD. So just to be fair, it's not something that's a unique advancement that's peculiar to array CGH, but it is part of the array CGH package of information.
Thank you.
Yeah, I see something. I see the light flashing away at me here.
Can I ask one more question? Because I think one thing that's confusing to us sometimes is understanding genetics when we're a carrier versus there's a chromosomal error. And from what I understand, you will be able to tell either way what's going on. But I understood you to say that when there is a chromosomal error, as opposed to you being a genetic carrier for something, that is likely to repeat as well. And so I became a little bit confused, and maybe I just don't understand, the filament's creating the chromosomal error versus I'm a genetic carrier for something.
You're right. Two different things. Let Dr. Munne handle that.
Yes, so there are two mechanisms in which you could have a baby that it's abnormal. One is they are inherited, so the couple is carrying each one of them the same genetic abnormality, although they could be perfectly normal. But if the two chromosomes or the two genes that are abnormal, they go to the same embryo, then you have an affected baby. Or it could happen that just the egg, because of aging and other factors, the chromosomes get misaligned, and then you would produce an abnormal embryo that is chromosomally abnormal. So two different things, but we use the same techniques, or different techniques, but the same method more or less to detect both of them. Right now, we need to know in advance if the couple is carrier of a genetic disease in order to look for them, because we can only look right now at a few genetic diseases at the same time in the embryo. For chromosomal abnormalities, we don't need to know. We can look at all of them. But for gene defects, we need to know specifically which ones to look for. That's why we want the couple to be tested, because otherwise we cannot do it. In the future, and probably in a couple of years, that won't be necessary, and we'll be able to check at the embryos directly. But right now, we still need to know if the couple has some genetic disease to be able to test it.
And I understood that you could do both, figure out if there's a chromosomal.
We could do both at the same time.
But what I didn't understand...
We still need to know what to look for. So you have to tell us, or we have to screen the couple to see if they have any genetic defects.
For the carrier part.
Yeah.
But with the chromosomal abnormality, you said that if that happens once, that there's very high likelihood that happens a lot or will happen again?
Yeah, that you have a higher chance of having more chromosomal abnormalities in the embryos. So for those, we don't need to test the couple. We just need to test the embryos.
Okay. Right. Thank you, gentlemen.
Thank you.
Thank you.